So we'll continue with our review of gene expression regulation. We left it here at how cells can control the degree of tightness in nucleosomes by chemically modifying the histone proteins in the histone core, such that if they acetylate them or methylate them, they will make them less positive, and therefore DNA will interact with them less strongly, and that will loosen up those nucleosomes, making the sequences in DNA more accessible, at least to those DNA binding proteins that need to read sequences of nucleotides in DNA. Then those, depending on their combination, would bring about or not the chromatin remodeling complexes that will ultimately devoid an area of chromatin from any nucleosome, such that RNA polymerase can come to a core promoter and initiate transcription. And we mentioned that these modifications are reversible. So for every acetylation, there can be a deacetylation. Despite of them being reversible, which may come across or sound, I think we had a bit of an interaction there. When we say reversible, that may feel a little bit like unstable or something that is a bit flaky and can go away on its own at any time, but that's not the case. So reversibility doesn't necessarily mean that it is going to happen for sure or in an uncontrolled manner. The reactions that deacetylate or demethylate histone molecules are active decisions that cells need to make. So these modifications are very, very stable, to the point that the decorations, you'll hear that language sometimes when talking about histone modifications, that histones are decorated through a series of specific acetylations and methylations at specific positions. So these decorations of histones can be inherited, not only from one cell to the next within an organism, but also between generations. When an organism places some of these decorations in their gametes, now these decorations make it to the next generation. When we talk about inheritance among cells, yes, it would be the cells that originate from cell division, from cell, let's say, mitosis. The daughter cells will inherit the same epigenetic modifications, those same decorations of histones that the mother cell had. And in more technical terminology, I know that I mentioned this on Monday, that what we really say is that the progenitor cell passes these histone modifications to the progeny, to the descendant cells. Okay? So any questions about any of that? And that's how we kind of wrapped it up on Monday. So from there, I wanted to discuss that when we talk about epigenetic modifications, you'll hear that term a lot, and you know, epigenetics became a thing as of more recently because of, you know, we're trying to more deeply understand its importance in regulating a whole bunch of processes. So when you hear about epigenetics really comes in two flavors. There is all of those chromatin or histone modifications that I just described for you, and then there is also DNA methylations. So I don't expect you to remember the chemistry, you to remember the chemistry of nucleotides by heart, but this is the nitrogenous base associated with cytosine. Okay? So down here would be the sugar and the phosphate that make up the letter C in DNA. It turns out that there are chemical reactions that eukaryotic cells can carry out, such that a methyl group is added to the nitrogenous base in cytosine. So we refer to those as methylated cytosines or methyl cytosines. Okay? It turns out that DNA methylation in cytosines usually takes place in what are referred to as CpG islands. And the name comes from the fact that when you have a CG dinucleotide, we don't write it out of convenience. We simply say CG dinucleotide, but the biochemical reality of it is that there is a phosphate group in between them. So that's why we refer to these as CPG islands. Right? So the enzymes that like to methylate DNA at specific positions, they prefer to methylate cytosines in these stretches of CG, 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 CG. Show of hands, who had heard about DNA methylation before? Some of you. If I asked you point blank, are DNA methylations associated with inhibition of gene expression or activation of gene expression? Inhibition. I don't think I caught your name. Oh, man. Emily. Where do you know that from?
Something like that.
Something like that. Excellent. We'll go with that. So you're absolutely correct. So these CPG islands happen in the promoter regions of genes, and they're usually associated with turning the gene expression off. I had it there on the screen, so much for a catchy question. That's absolutely correct, right? So these DNA methylations are typically associated with inhibiting the expression of genes. Now, let's see if I have it here on the slide. So let's imagine now that there is these two nucleosomes, right? In the before state, if you will, the nucleosome is pretty relaxed because the histone tails have been acetylated and or methylated, so they've been neutralized and they're not interacting with DNA so tightly. And now we have a whole bunch of DNA methylations that add methyl groups to a CPG island. So we'll have a lot of cytosines that are methylated in a row. My question to you now is, how can these turn off gene expression from a sort of a biochemical point of view? How can the methylation of cytosines turn off gene expression? So in this case, you're saying that the methylation of DNA is modifying the charges or the properties, and how would that impact on gene expression mechanistically? What could happen to this region of DNA? Loosening or tightening. So if we're saying that the gene expression of those that are methylated is normally off, would you expect tightening or loosening? Loosening, make it more available. Tightening, defend your position.
I forgot the name. It starts with an A.
The acetyl group.
Group, you have to tighten it up to turn off.
To turn off the gene expression.
If I remember.
Let's see. What?
No, what is that? Yeah, the tightening was like that.
Do you see it now? So normally we associate tightening of nucleosomes to reduce gene expression. Why? Because we're making this DNA now less available for those transcriptional activators that are necessary to recruit the chromatin remodeling complex that would devoid this area of nucleosomes such that RNA polymerase can go to work. Hairpins, and we will talk about hairpins incredibly soon. Hairpins are structures that originate in single-stranded molecules, right? So we normally leave the hairpins to RNA molecules, not so much DNA. Because for hairpins to form, you need those nucleotides available in a way to engage in base pairing with nucleotides within the same molecule, whereas these guys are base pairing with one another across both strands of DNA. Yeah. All right, so to recap, DNA methylation is normally associated with turning expression of genes off, and a mechanistic model might be that there is some sort of change in the property. I think, Josh, you mentioned like charges maybe that would tighten these nucleosomes such that now they are less available to proteins that read sequences in DNA. You guys are using your heads, right? You're thinking logically about this. Except that I need to throw out this idea now. And that is, what charge let's go back one slide. What charge in the cytosine is being neutralized here by methylation? we're saying that this thing is associated with a positive charge and now is no longer associated with a positive charge? Is this hydrogen positively charged in DNA? It's not. So yes, DNA is a charged molecule, and we discussed last Monday that it is negatively charged. Yeah.
I was thinking that the hydrogen is less bulky than the methyl group, so does that have something to do with that?
Okay, excellent. So the hydrogen is less bulky. So the methyl group, what could it do to DNA? If you stick in something that is bulkier, so let me take a step back. What will that C be pairing with in DNA? A G, correct? So what could that methyl group be doing to the CG base pair?
Maybe blocking it from being able to bond correctly to the gene?
Right? Again, a reasonable idea. The one element that you didn't have to have, but that comes from understanding a little bit more the chemistry of DNA, is that hydrogen is actually not involved in base pairing, right? Yeah, it's mostly these three. So these three, that's going to be offering a hydrogen, that's going to be accepting a hydrogen, and accepting a hydrogen. Right? Good idea, but no. Just a little confusing, right? For the histone molecules and their acetylation or methylation was pretty clear cut. We're modifying them chemically such that they're no longer as positive as they naturally are. So they're going to be interacting with DNA less strongly, and now the nucleosome loosens up a little. With methylation of cytosines in DNA, there is no clear-cut biochemistry of what might be going on. That confused scientists for a while, and it turns out that what happens is these methyl or methylated cytosines, and I'll go back to what you were saying before, Josh. These methylated cytosines, you used an interesting term. You said it changes their properties. And I could totally agree with that. It's not changing their charge, but it is in a way changing their landscape, right? Their bulkiness. And for us, it's just maybe one more atom or a handful more of atoms. But for proteins that can read sequences in DNA, it makes a world of a difference, right? So there will be proteins that don't care much about unmethylated CpG islands. If they see a stretch of regular CG, it's almost invisible to them. They don't care about those. The second you present them with a methylated CpG island, where you have CG, but every one of those cytosines is methylated, now all of a sudden they can see that, they can interact with that biochemically. And what has been shown is that many of those proteins that can interact with methylated cytosines can recruit deacetylases that will take out the acetyl groups from the histones, bringing the nucleosome back to their tight conformation. Right? So we started out from a loose nucleosome that was fully accessible to transcriptional activators that could have brought the chromatin remodeling complex to start expressing genes in that region. But now through methylation of the CpG island, we've created a landing pad for these molecules that can read them and say, hey, we should bring a histone deacetylase, shave off all of these acetyl marks from the histone molecules, will make them positive again, those reversible reactions that will make those histone molecules positive again. And now DNA and histones will love each other much more strongly once again, creating that tight nucleosome conformation that makes the DNA molecule within that nucleosome a lot less accessible to activators and repressors. Right? So that is how, and this is what the story that you may have heard before, that is how methylation of promoters is associated with silencing of genes, because for as long as those promoters remain methylated, those promoters will most likely exist in these tight nucleosome conformations that will not allow the transcriptional activators that you normally need to recruit RNA polymerase to get there. Yeah, all right, so these are both what we call collectively chromatin modifications. So you'll see references to regulation of gene expression through chromatin modification. You can refer to either side of chromatin. Remember that chromatin is the combination of DNA and proteins, more specifically these histone octamers. So you can modify chromatin by either modifying the histones or the DNA. None of these, you should bear in mind that none of these includes genetic mutations. That's like a separate category. Genetic mutation is a change in the letter, in the identity of that nucleotide. And that, of course, can happen too. And that, of course, will be inheritable, right? But we're not talking about that. We're talking about these epigenetic modifications that can bring about some regulatory information, some level of regulatory control to the expression of genes that have nothing to do with changing the sequence of nucleotides of letters in DNA. Okay? Cool. So, let's imagine now that the cell loosened up the chromatin, went ahead and modified a bunch of nucleosomes through histone modifications, such that those nucleosomes became less tight, allowing for the recruitment of the chromatin remodeling complex that will remove nucleosomes from a given region of DNA, exposing what the cell really needs to start the transcription of a gene, and that is the promoter region, right? And when we talk about a promoter region, you guys may remember that promoters are generally subclassified into core promoter. That's where the actual action of assembling a transcription initiation complex and ultimately RNA polymerase will bind DNA and start transcription. And then what we call a regulatory promoter. And the reason we normally subclassify them into those two is because the core promoter normally does something that is kind of always the same. It's always very predictable. The transcription initiation complex assembles at the core promoter through the binding of a series of proteins that are always the same. Some of you, particularly if you took my genetics, you may remember the TF2 family of proteins. There is TF2A and B and C and D all the way through H. And they will start piling up in these core promoters, and they will use consensus sequences in these core promoters that are the same across genes. There is the TARA box in many genes. You may have heard of that. There is CG-rich boxes. Those are the ones that normally get methylated and in a way obscure my methylation. There is CAAT boxes. There's a bunch of consensus sequences that these core transcription factors use to assemble at the core promoter. And once they're all in place, they can summon or they can recruit RNA polymerase. So when that happens, RNA polymerase will separate the two strands of DNA and initiate transcription. But that is not full transcription. Whenever RNA polymerase is doing this at its basal rate, we refer to that as basal transcription. And it turns out that RNA polymerase is a little kind of lazy. If RNA polymerase is recruited to a core promoter, the next one may not be recruited until a little later. There isn't that many RNA polymerase molecules going around in the nucleus, and they're finding a pre-assembled transcription initiation complex may take a while. Even if it is in the scale of seconds, in molecular or molecular genetic scales, that's an eternity, right? So when you leave the transcription initiation complex and RNA polymerase to express genes at their own devices, that's going to be just basal transcription. Yeah, you're going to generate an RNA molecule every once in a while. If a cell really wants to express a gene at very high levels, it uses a series of facilitator proteins. So there is transcriptional activator proteins, there are co-activators, and more importantly, there is a big protein complex called a mediator. And think of these as scaffolds that RNA polymerase finds it a lot easier to bind to than the transcription initiation complex on its own. So it's almost like whenever RNA polymerase sees a mediator at a core promoter, its rate of binding and initiating transcription at that core promoter will be exponentially increased. So that's how we go from basal transcription to regulated or full transcription. And it can go both ways. When a given area of DNA is exposed, you can have transcriptional activator proteins, but sometimes you can have some transcriptional repressors. Those are proteins that are saying, I know that you want to express this gene at some point, but not yet. We need to wait for specific signals. We need to wait for specific, some sort of sign that it's okay to express this gene now. Until then, I will occupy a space in the regulatory promoter, and I will not allow RNA polymerase to be recruited, even though the area is free of nucleosomes, even though the transcription initiation complex may be partially to fully assembled. I'm not letting RNA polymerase do its job until something tells me that I should leave, and now you can proceed with transcription. All of that happens in the regulatory promoters, shown here in like some sort of orange color. Now, I keep saying that transcriptional activators and repressors bind these regulatory promoters. How do they know where to bind? How do they know that a given transcriptional activator, how does it know to go bind a specific regulatory promoter? Yes? Motifs. So motif, a different term for that, like a sequence, exactly, that's it. Motif is a sequence that kind of repeats itself. So it's a term that I guess molecular geneticists borrow from music. But it's the idea that it's a specific sequence of nucleotides that repeats itself in different genes, right? So we call those DNA motifs. That's exactly correct. So what happens is these regulatory promoters are actually collections of these motifs that are bound by specific activators or repressors, and that are in turn activated by signals that come into that cell. So a given transcriptional activator may have a target sequence in a given promoter, but may bind to it or not, depending on the state the cell finds itself in. Right? So that's where the regulatory gate takes place. So let me take a step back and spend 1 second here. So the idea is, and this I suspect we're going to find a lot of in our exploration of the literature. The idea here is, when will RNA polymerase start transcription? Well, when there's a bunch of transcriptional activators bound to their corresponding transcription factor binding sites, their TFBSs. Okay, cool. We've just kicked the question one step back, right? So now I could ask the question, well, when does a transcriptional activator bind its transcription factor binding site? Well, when the transcription factor binding site is present in a promoter. And then you might say, okay, when is a transcription factor binding site present in a promoter? That's a question for evolution. Those transcription factor binding sites evolved to be there because it was useful for the organism to have those transcription factor binding sites in their promoter. But there is something missing. And that is, we kept kicking the question back, but we're still not addressing the fact that none of this is useful if the transcriptional activators will be binding to their transcription factor binding sequences all the time. If they're binding to their transcription factor binding sequences all the time, then all genes are expressed all the time. And that's not good for cells, right? Because if all of the cells expressed all of the genes all the time, there wouldn't be any diversity in function. That's one way to look at it, right? That if muscle cells expressed all the same proteins that neurons expressed and vice versa, how would they be different? How could they do different things? Another way to think about this is, you know how we talked about these reversible reactions? That the cell at some point might make a decision to acetylate a histone, but then at some later time, it may decide to deacetylate it. What would happen if both the acetylase and the deacetylase are expressed simultaneously? Then that histone would be shuttling back and forth between acetylated and deacetylated all the time. There would be no regulation in the state of that nucleosome, right? So those are two arguments for why it is so advantageous for cells to not express all their genes all the time. But everything we've done so far is kick the question back, right? We say, well, not all genes are expressed all the time because RNA polymerase is not binding to their core promoters all the time. Oh, cool. How does that happen? Well, because RNA polymerase is not brought to all core promoters at the same time. Oh, great. How does that happen? Oh, because not all the genes will have all of the transcriptional activators that they need at any given point, right? And we kind of stop there because what do you mean they're not brought to DNA all the time? The transcription factor binding sites are written in DNA. They're there all the time. So why are transcriptional activators not binding to their regulatory promoters all the time? So the comment here is maybe they don't want, I'm paraphrasing, but they don't want to waste energy. Am I paraphrasing correctly? I agree with you. That is, if you will, the adaptive value of all of this. So why express all genes all the time if it's not going to do anything good? Exactly. So the saving energy is, if you will, a bonus. But the question was slightly different. The question is, if those binding sites are there all the time, what makes an activator see them or not? Excellent. So that's one level, if that protein is present or not. So the binding sequences are there all the time, but the activator itself may not. A neuron may express certain activators that muscle cells don't, and vice versa. Right there, you have a mechanism by which neuron cells can express certain genes that muscle cells can't, and vice versa. That's a good one. What else? A specific substrate. What do you think of? Like an activator and what's present? Tryptophan, right? Like these molecules. And you're absolutely correct. So sometimes activator proteins are present in cells, but they cannot bind their binding sequences yet. They need a molecular partner, right? We call those inducers. And to some of you, it may bring some memories of things like the LAC operon and TRP operon, if you studied them before. This idea that proteins that bind DNA will bind DNA depending on what shape they exist in. And the shape of those proteins can be modified by these molecules that interact with them. So some of these activators may require some sort of cofactor or inducer to let them morph into a version of themselves that can bind these binding sites. Yes?
I was just saying the same thing, that the activated protein goes through a conformational change.
Conformational change. So
Allows them to bind to the active.
beautiful. And we just talked about inducer molecules or cofactors that will interact with these activators and make them go through that conformational change. What else can force a protein through a conformational change? We just mentioned that. So the binding of a different molecule can change the shape of a protein, for sure. What else can change the shape of a protein?
Different ions?
Different ions? Okay, ions could be these molecules that interact with them, but close, very close to the idea of ions. The shape of the protein, I'll take that. There is a few examples, so just so that we're on the same page. So the comment here is abiotic factors like temperature, pH, in a way ions, pH, ions go hand in hand. So those things can change the shape of proteins. And one example that immediately comes to mind, there are genes that respond to sudden increases in temperature. Promoters that can detect a sudden increase in temperature, we say that are heat responsive, right? So there are a series of transcriptional activators that change shape when it gets 1 or 2 degrees hotter. They will change shape, and now they can bind to those promoters and activate the expression of genes that will most likely allow those cells to tolerate that heat stress. Well, I suppose you could say that the transcriptional activator in its new shape, it's catalyzing the initiation of transcription. I could take that. But I think maybe I don't want to put words in your mouth, but maybe what you're thinking relates to what Ali was saying about enzymes, right? And you're so close.
Would a correlated protease not be available?
The one?
A protease.
A protease.
Correlated protease to whatever protein.
So what does a protease do?
It can modify, chop up, reduce.
Chop up. That's the key word. So a protease will chop up. So so that we're on the same page, proteases are enzymes that will cleave off or chop up a protein. Typically, they will cleave off a fragment of them. Are those reactions reversible?
If there's something else.
They're not. They're important in their context, but they're rarely reversible. If you chopped off a piece of a protein, I don't know of any D protease that will add it back or anything like that.
Wait, can you repeat the question again?
Yeah, the question. So we mentioned molecules that can bind to these activators, that can be inducers, right? Including things that like ions that may, by interaction with these proteins, change their shape. Abiotic factors that can, like temperature or pH, change the shape. And then we opened up the can to enzymes. And my question was, what can enzymes do to these activators? And one with the comment that we were just discussing was, maybe if that enzyme is a protease, the protease will cleave off a bit of that activator and will make it now bind the binding site. The issue with that is that those chemical reactions, those enzymatic reactions are not reversible. So once you chopped off a bit of the activator and the activator is bound to the promoter, you can never turn that gene off because you can't add a piece of peptide to the activator. I have several hands up, so I'll go in order.
So you mentioned something
Something like reversible. Ligands would be like we were discussing before, inducers, molecules that are not really modifying chemically the protein, but you have it there. Exactly. Phosphorylation. You were going to say phosphorylation? Yes? You promise? Pinky promise? And by the way, I got to get your name so I can write down. But exactly. So phosphorylation is a classic mechanism by which cells can turn on transcriptional activators. The transcriptional activator is sitting in the cytoplasm, or maybe inside of the nucleus, in its unphosphorylated form, right? And an enzyme that we normally call a kinase will come, add a phosphate group chemically. Now that phosphate group has a bunch of negative charges associated with it, and the transcriptional activator will have to adapt to that new reality of having a bunch of negative charges associated with it. What does it do? It kind of shapes, shape shifts and adopts this new conformation that will make it able to bind the promoter. How do you turn that thing off now, right? The same argument. How do you turn it off? Well, just like there are kinases that phosphorylate proteins, there are enzymes that take them out, take the phosphate groups out. Do we know what those are called? Transferase we leave for things like they transfer a bigger molecule into a protein. So, Phosphatases, that's right. Not very original, the name, right? So protease, chop off protein, or a piece of the protein. Phosphatase, chop off the phosphate. But that's exactly what it is. Phosphatases can dephosphorylate these activators, and by doing so, turn them off. So in a way, all of this to say that these reversible conformational changes in activators and repressors are what bring about the regulatory gate, the logical gate in all of this. These reversible changes in these activators and repressors, or the fact that some of them may be expressed or not by a given cell, are the ones that interpret the internal and sometimes secondarily the external environment of a cell. Why? Because if a cell is getting a little too hot, those heat shock promoters will be bound by proteins that shapeshift with heat. If the cell is receiving a lot of insulin, now there's going to be some transcriptional activators that are phosphorylated by the insulin signaling pathway. Ooh, how does a cell know if itself is a muscle cell? It has to do with its developmental history. So, and it relates back to what Sebastian was doing. So, saying. So the idea is a muscle cell didn't just come out of nowhere. It was first a pre-muscle cell, like a muscle precursor cell. Before that, it may have been a mesodermal cell. So, and all along the way, it started. And here's where it gets interesting. You can lower your hand. I know that you have a question,
Sorry.
so that you don't get an shoulder cramp. It has to do with the fact that along its developmental history, it will permanently turn off the expression of certain genes. So once a cell committed to mesodermal fate, it says, well, I'm not going to need any of the genes that those ectodermal cells and endodermal cells normally express. I'm going to turn them off permanently. How do cells turn genes off permanently? What cells do will super compact a certain region into heterochromatin, right? So that's when an entire region of the genome is like stored away for good, right? So throughout the developmental history of a given cell lineage, they will stop expressing certain factors and start expressing others. So at any given time in, let's say, modern life, or in the current day, a cell will know what, quote unquote, it is by the collection of activators and repressors that it expresses, that it has. Whether or not it's using them or not, it may depend on external and internal conditions. Is that cell properly nourished? Is that cell under metabolic stress? Is that cell a little old? Is that, and so forth. So all of those things would translate into these things dynamically binding or not to their binding sequences in regulatory promoters. But at least they're there, right? They're there to ask the question. So the muscle cell will ask the question, am I well nourished? Am I young and healthy? Am I not under any kind of oxidative stress? So if the answer to all of those is yes, all of these guys are going to be activated, bind to this thing, and now the gene will express. And last but not least, this is kind of important, and I'm sure that most of you, if not all of you, heard of this before. These facilitating of the formation of an initiation complex and the initiation of transcription by RNA polymerase can be done locally through those regulatory promoters that we just discussed, or distally, at a distance, by similar collections of transcription factor binding sites, TFBSs, that happen far away from the gene in regions that we call enhancers. Okay? So enhancers can activate gene expression from thousands of base pairs away from the actual gene that they control. And that is possible thanks to what we call DNA looping. So DNA is a relatively flexible molecule, right, that can loop in space, can turn in space, effectively bringing an enhancer that may be thousands of base pairs away, right? So the distance between the actual core promoter and the enhancer itself may be thousands of base pairs, but through DNA looping, the enhancer comes in very close physical proximity to the core promoter, and now the transcription activators that are bound to that enhancer can do their job as if they were right next to the core promoter. Close physical proximity to the core promoter. That's ultimately what matters, is that the enhancers are close to the core promoter so that they can bring more RNA polymerase and ramp up the transcription of that gene. So what do we know about enhancers? Enhancers can control gene expression from thousands of base pairs away. They can be either upstream or downstream of a gene. So here's, we're representing the enhancer here as this orange box. And this enhancer can stimulate the expression of these genes, but it can also stimulate the expression of these genes. And when you think about the relative position of that enhancer to where the gene starts and in which direction it goes, it's actually downstream of those genes, right? It's a little easier to see it here with the one on the left. So the enhancer is, quote unquote, after the gene. Right? Here's where the gene begins, and it goes in that direction, and the enhancer is after it. And for the one here on the other strand, same thing. So think about this gene as starting here and then going in that direction. This thing is downstream of it. How can that happen? Again, looping. Because just like the DNA string can loop on the same plane, it can also kind of twist in the process, and it positions itself as if it were a regular promoter, regulatory promoter, I should say. Yeah? So it's that flexibility in the DNA string that allows enhancers to operate from a distance and in a way that is relative position independent. And last but not least, this is a little less obvious, so I wanted to spend a second discussing this. One of the, I guess, conundrums, you could say, when enhancers were first discovered was, well, if enhancers can operate at a distance that is in the thousands from a core promoter, right? If you have an enhancer anywhere in the genome and you go, let's say, 5,000 base pairs in one direction and 5,000 base pairs in the other direction, that enhancer should activate the expression of all the genes that you find within that region, right? So in that span of 10,000 base pairs, that enhancer should turn them all on. And experimentally, people were finding that that's not the case, that within a region of influence of a given enhancer, that in this case we know is at least from here to here, there were some genes that, yes, were activated by that enhancer, but some genes that weren't. So how come? Too tightly packed. Excellent. Not what I have in my next slide, but yeah, I'll take it. So maybe this particular gene is in tight nucleosomes. The histones around these promoter are totally deacetylated, totally demethylated, and that DNA and those histones are hugging each other so tightly that no one can come in. I like that. It's an interesting thought. So the idea here is, once the activators have assembled at an enhancer, what they do to communicate with RNA polymerase, that doesn't require any more activators. It all goes through the mediator. So the point is, once an enhancer is, let's say, turned on and, for lack of a better term, let's call it hot. Once an enhancer is hot, what it's doing is interacting with the mediator complex. So theoretically, anywhere where there is an open core promoter, it could bring the mediator, and next thing you know, RNA polymerase will love that core promoter and keep transcribing. But it's really close. You mentioned transcriptional activators, right? What's the opposite of the activators? The repressors. And more often than not, when you see a gene that is theoretically under the influence of an enhancer, and provided that it's not in tight nucleosomes, what happens is that when you go look in the regulatory promoters of those genes that are activated by the enhancer, you see a bunch of binding sites for activators. So it all makes sense, right? So you have some of the activators, you have the core promoter. Those binding sites for the activators may be occupied or not, doesn't really matter because the enhancer is there to turn that gene on. But when you look inside those genes that are within the area of influence of an enhancer but are not being turned on, more often than not what you see are binding sites for repressors that are telling those genes, yeah, I know that that enhancer is hot, but we're not expressing this gene now. And how those cells make that decision, again, from a biochemical point of view, it all comes down to those genes having the binding sites for those repressors, and for those repressor proteins to be in a shape that allows them to bind DNA. When those two things check, now you have a repressor bound to the core promoter or near the core promoter, and it doesn't matter if there is an enhancer coupled to a mediator complex, that gene is out of sight. Even though through DNA looping, this hot enhancer could reach its core promoter just fine, okay? Why am I doing all of this? It's not for you guys to remember necessarily, it's just to kind of expand a little bit the broad range of possibilities of how things start piling up in a way, in the sense that we have presence or absence of binding sites in promoters, but also expression or not of the different activators and repressors, and then chemical modifications or inducers interacting with these proteins and so forth, right? So it's all of these boxes in a way that a cell needs to check before it decides to express a gene. Yeah? I will answer this way more often than not. Yes. So what happens is cells, now I'm going to borrow your idea of economy. Cells don't like to express a gene more than they should. Evolutionarily, they learn to be very frugal and economical about everything. That includes gene expression. So if I express the gene to the point that what I wanted to do, I'm doing, why express more of it and waste these precious resources? So what happens is, let's say that, let's take on one of these that is activated by the enhancer, this guy here. So this gene expresses protein X, doesn't matter. Protein X contributes to process Y, right? More often than not, process Y will feed back, usually negatively, in some sort of way through, for instance, dephosphorylation of some of the activators, through, for instance, active degradation of some of those activators, through, for instance, putting out a whole bunch of molecules that will interact with those activators, make them change shape such that now they fall off of DNA. All of those things could happen such that now this activator that was bound to this enhancer and activating the expression of that gene now falls off. Now, you guys are probably very used to the idea of allosteric feedback inhibition in enzymatic pathways. That's how most of us learn about this concept, right? So you have a series of reactions and the last product can go bind the first enzyme and turn it off. Here it's usually a little more complex than that. So it's usually a result of the actual process that this gene is contributing to. So the whole process is what feeds back. So it's not that this protein will go and physically interact with the activator, or it's not that this protein, even if it is an enzyme, will produce the metabolite that will go and inhibit the activator. Generally, it's a much more indirect process by which there's several steps that need to take place before this thing gets turned off. What came after transcription? If you guys remember, a series of post-transcriptional regulatory mechanisms, right? And what cells, eukaryotic cells, do to RNA transcripts that they got off of DNA from DNA protein coding genes. Much of what we're discussing now is about DNA protein coding genes. Is they need to cap them in their 5' end, as we discussed before, to protect them. They need to polyadenylate them in their 3' end. More often than not, there's going to be splicing of introns out, and that opens the door to a phenomenon that we call alternative splicing that I think we'll discuss today. And then nuclear export to the cytoplasm. Because eukaryotic mRNAs are produced in the nucleus, but they're translated in the cytoplasm, so they need to be transported into the cytoplasm first. Right? Turns out that 5' capping and nuclear export to the cytoplasm are relatively universal, meaning that all protein coding genes will go through the same, or all maturing mRNA molecules will go through the same. So it's not necessarily a major regulatory point. If all genes go through that, then there is no way for cells to use those as mechanisms for saying, yeah, I'd like to express this gene or not. Does that make sense? Yeah. So we're not going to discuss those in further detail. When we talk about polyadenylation sites in genes, we mentioned the other day that RNA polymerase creates these super long transcripts. Somewhere in the sequence of those transcripts, there is a polyadenylation signal that tells enzymes in the nucleus, hey, let's truncate this mRNA molecule here, and let's add a bunch of adenines to the 3' end of this molecule. Right? So that's what eukaryotic cells do with their mRNAs. So those signals sometimes happen only once in a primary transcript, or what we normally refer to as a pre-mRNA molecule. But sometimes, every once in a while, you will have more than one polyadenylation signal. And now cells can choose to use one or the other. Right? And anytime that a cell has a choice for one or the other, that's a regulatory opportunity. Right? So every once in a while, you have, let's say, these two polyadenylation signals, and it'll be random. So half of the transcripts will end up with one short isoform and the poly A tail sooner. And the other half of the times will be longer when it starts from the second polyadenylation signal. But other times, cells can actively decide which one they will use. Now I have a question for you. Alternative poly-A sites, like the ones shown here, rarely change the CDS, meaning they rarely change the sequence between the AUG start and the stop codon. Almost by definition, they tend to happen after the stop codon, right? What's the point of polyadenylating an mRNA before you reach the stop? That cell would be making incomplete proteins, right? So there are some exceptions, but for the most part, these alternative poly-A sites happen after the CDS. So how can these two mRNAs give rise to different effects in the cell? Different proteins being expressed, and therefore different flavors in the expression of a gene, as Oscar was saying on Monday, right? So different phenotypes. How can these two mRNAs, that one shorter than the other, but both have poly-A tails and the same protein coding message, give rise to diversity? So you're saying one is longer, it might affect how the mRNA is expressed, and how could that be? If the message to be translated is between those two, and it's the same in the short mRNA. Loop back. Okay, I'll take that. Loop back. So you're saying that maybe this longer molecule is somehow looping back and messing with the translation or something like that? Less stable. All right. So it's more prone to decay. I like that. So a slightly more complex version of the same thing. So instead of looping, it starts forming hairpins. I like that too. Yeah. Exactly. So the truncation happens once when the poly A tail is added, but from there on there is no further truncation. And if the poly A signal is downstream of the stop and the whole coding message has been spared. So there is no like chewing into coding sequence. If I'm interpreting you correctly. Well, so the question was, how come that these two messengers with the same message for making a protein can give rise to different outcomes for the cell if they will express the same protein? I thought that we said mRNAs, they don't do anything by themselves. What they do is they bring a message that the cell turns into a protein through translation, and it is the protein that does things, right? So how come that these two mRNAs, different mRNAs expressed by cells at different times or under different conditions, can do something different when they're both expressing the same protein? So during nuclear export, the cells will check a 5' cap and a poly A tail. They're not going to check how long the RNA molecule is in between them, because the mechanisms for nuclear export know that some mRNAs are super long, some of them are shorter, so they can't really make decisions based on that. So when it comes to export these two isoforms of this particular mRNA, the nuclear export machinery won't be able to tell them apart. You guys had it, really. So with more of this sequence that happens after the stop codon, which we called, you guys remember what name we gave to that region in an mRNA? It's the 3' UTR, right? So with longer 3' UTRs, there is you're basically creating a possibility for funny hairpins to form, for some weird looping that will slow down maybe translation. And then one that I didn't hear, but luckily I have in the next slide, you're creating room for potential targets for degradation. And this will bridge us into something that we'll discuss a little later. But the idea is that longer 3' UTRs may carry in them sequences that make those mRNAs targets for degradation by the cell itself. We mentioned briefly last week these small non-coding RNAs, many of which have regulatory functions via the active degradation of specific genes. So that's exactly what we're talking about. So the small RNAs that target mRNAs for degradation do so by finding in them specific sequences. Just like proteins find in DNA binding sites for them to bind DNA at specific locations, these small non-coding RNAs that degrade mRNAs do so by binding to specific target sequences in typically 3' UTRs of genes. So the longer the 3' UTR, the more of these target sequences that you may accumulate. And essentially what cells are doing then is make these longer isoforms sensitive to degradation by specific microRNAs or siRNAs. The mechanisms of that we'll discuss a bit later. Okay? So alternative polyadenylation is a regulatory mechanism because it can create some funny secondary structures and because it can open up the door for additional target sites for degradation. Yeah, no, that's an excellent question. So if you want to destroy the message, why not put those targets for degradation here, right? The thing is that if you have the targets for degradation here, you may be messing with the protein or the CDS, right? So having a target for degradation here might mean that you need to change the protein code. You don't want to do that. If you have the targets for degradation in the 3' UTR, you're not messing with the code, and the whole thing gets degraded anyway. So even though you have the target outside of the coding region, you're killing the expression of this protein as a whole. We'll see that in a second. Just hold that thought. What a beautiful question. When we say that opens up targets for degradation, one might think, the way that I said it, maybe, sorry, is that the second that we open these targets, this thing will get degraded. The way that evolution sees it is, well, no, hold on a second. The way that we open up these targets, there is the potential to be degraded. So the difference between this isoform and this isoform, the shorter one will translate until this mRNA decays through natural means, right? Through degradation or attrition of the poly A tail. Maybe they lose the 5' cap, right? With all these ribosomes coming in and whatnot. So this thing, all molecules have some sort of lifetime, and for these short isoform, we're kind of depending on the half-time of this molecule. For the longer one, what evolution does is it says, I'm going to have those bullseyes ready, and if the cell receives a signal that says, stop it with this protein, then I'm ready to degrade it quickly. The presence of those targets doesn't necessarily mean immediate degradation. If it meant immediate degradation, I could not agree more with you. It would be a phenomenal waste of energy to express this longer RNA to then degrade it. But when longer 3' UTRs expose new targets, it's not a given that they will be degraded. It's just now they are degradable if needed. All regulatory for the expression of these protein. So first the cell decided if it's going to transcribe this gene or not. Now it's deciding if it will keep a short mRNA or a longer one. So far it's just about decisions that the cell is making along the way. All right, so let's hold that thought about the targets for RNA degradation for a second, because I want to discuss also alternative splicing. So coming in this class, you guys knew about splicing as a phenomenon. I remember when I asked you to describe a eukaryotic gene, one of the first things that I heard was exons and introns, right? So we're all on the same page that in DNA, most eukaryotic genes will have sequence that get transcribed into the RNA and that we divide into exons or introns depending on whether or not we'll find those sequences in the mature mRNAs. Any sequences that we don't find in the mature mRNA have been spliced out, and we call introns. So let's imagine this pre-mRNA molecule that has the exons and the introns as shown. So the color boxes are the exons, the gray boxes are the introns. After splicing, the mature mRNA will have only the exons, not the introns. A slightly different depiction of the same thing when you're drawing genes in DNA. The way that you show them, and you're going to see these types of cartoons in papers all the time, the way that you show it in DNA is all of this is DNA sequence. I know. But what we do is we indicate with boxes the exons and with connecting lines the introns, and these carrots on top of the introns simply indicate these things are going to be cut out and spliced out. Right? So at the end of the day, it's two different depictions of the same process. Many genes are going to have what we call constitutive splicing. That's the simplest, the most straightforward to understand. All of the introns get spliced out, period. But more often than not, it's calculated upwards of 90% of eukaryotic genes. Oh, and I was going to mention that mechanistically speaking, the way this happens is in between exon-intron boundaries, there are these consensus sequences that we refer to as donor sites on the 5' end of an intron and acceptor sites on the 3' end of an intron. And then somewhere in the middle, there's a sequence that we call the branching point. And what happens is nuclei have machines, huge machines, called spliceosomes that can read a donor side, they can read an acceptor side, they can read a branching point, and they will mediate a series of catalytic chemical reactions such that both ends of the intron are cut out, right? Then there is a branching reaction by which the donor side gets chemically attached to the branching point, forming these funny lariat structures that are immediately degraded by the nuclei because the nuclei know that these lariats mean introns. So it knows it needs to degrade those things because they're useless. Yeah? And in the process, both ends of the both ends of the exons, the preceding exon and the subsequent exon, get joined together, again, through a catalytic reaction that will create a phosphodiester bond between them. So that's how introns are spliced out. In constitutive splicing, what I drew in this box here happens within each of those introns, because the spliceosomes can find and read and use all of the donor sites and all of the acceptor sites. But what happens in upwards of 90% of eukaryotic genes is that there is some form of alternative splicing. That is when the donor sites and acceptor sites that spliceosome use come in different combinations, such that, for instance, this constitutive splicing that we had before now turns into exon skipping. We can imagine a spliceosome that saw the donor site from the first exon intron junction, didn't see the acceptor site from the first intron, but saw the acceptor site from the second intron. So what does it do? It takes the whole thing in between out. So effectively, what the cell did was skip an exon. Right? So in constitutive splicing, we should have seen 4 exons. When there is exon skipping, there is only 3. There are cases of mutually exclusive exons. So the combination, the shuffling of exons together can change from one cell to another, or within a cell from different states. Am I properly nourished, or am I starving? Am I receiving a hormone signal, or am I not? And then alternative splice sites that will make some of the exons be shorter or longer in the mature mRNA. So all of these things boil down to how spliceosomes see these exon intron boundaries, and whether they use them all or they may skip certain ones.